Have you ever wondered how one of the world's oldest and most cultivated crops could suddenly cause massive blood cell destruction in millions of people? It's a crazy thought, right? Well, welcome to a deep dive into the fascinating and, honestly, sometimes deadly world of toxic phytochemicals.
Okay, let's dive into this, because whether you're a food technologist, a toxicologist, a forensic scientist, or a clinical chemist, we are all actually looking at the exact same phenomenon here. Today we're exploring plant defense mechanisms. This is nature's very own chemical warfare, specifically designed to deter, disrupt, and sometimes completely destroy. We have 5 incredible case files on our agenda today. We'll cover everything from hijacking the thyroid and collapsing cellular defenses, to motor neuron meltdowns, dietary cyanide, and finally, a lethal lectin. We are going to trace the chemistry all the way from the soil straight into cellular pathology. So let's open case file number 1, hijacking the thyroid. We're going to talk about goitrogens. Now, before we look at the actual chemistry, here is a really fascinating historical fact for you. Did you know that the fashionable high, stiff collars worn in 19th century Europe were actually a fashion trend specifically designed to hide prominent goiters? It's true. Now, speaking directly to the analytical chemists with us today, the mechanics here are incredibly precise. The thyroid gland naturally concentrates iodine using a sodium iodide symporter, or NIS. Certain plant toxins, like thiocyanate, directly inhibit this exact transporter, while other substances, like goitrin, block the organification process entirely. It is a complete biochemical hijack.
So where exactly do these goitrogens come from? Well, they are extremely common in our environment. We see progoitrin in cabbage and rapeseed, thiocyanate from cassava, and polyphenols in millet. But here is the absolutely crucial point. It's how these act synergistically with iodine deficiency. Think about this. Public health efforts today strongly encourage us to reduce our dietary sodium to prevent cardiovascular disease. But by reducing our intake of iodized salt, these natural goitrogens are inadvertently causing a global resurgence of goiter. It's a massively complex public health challenge.
Moving right along to case file number 2, the cell's collapsing defenses. Let's look into phavism. Now, what's really interesting about this condition is that when susceptible individuals are exposed, the clinical symptoms are incredibly rapid and severe. We are talking about sudden jaundice and hyperbilirubinemia, especially in children. And the source of this toxicity? The common fava bean. Fresh fava beans contain very high levels of the glycosides vicine and convicine. Once in the gut, these get hydrolyzed into divacine and azoramyl. You can think of these as aggressive pro-oxidant saboteurs that get released directly into the bloodstream.
But why does this only affect certain people? Well, let me use a molecular analogy for the pharmacologists out there. Think of the enzyme G6PD as the red blood cell's financial reserve of reducing power, which it keeps in the form of NADPH. The fava bean toxins cause high-capacity redox cycling, which produces just massive amounts of reactive oxygen species. If a patient has a genetic G6PD deficiency, their cells simply cannot replenish their NADPH. The cell faces sudden oxidative bankruptcy. The structural proteins cross-link, the cell membrane collapses entirely, and the red blood cell is destroyed by the spleen.
All right, case file number 3, motor neuron meltdown. This brings us to neurolatherism.
Neurolatherism is an ancient and frankly tragic neurodegenerative disease linked directly to the Lathyrus pea. Because this hardy crop can survive extreme droughts or floods, it often becomes a desperate food source during severe famines. Biochemically, the toxin hidden in this pea is an amino acid derivative called ODAP. For the toxicologists, ODAP acts as an aggressive chemical weapon that completely overloads the AMPA subclass of glutamate receptors. This triggers prolonged calcium influx, massive reactive oxygen species production, and causes the specific death of Bett's motor neurons. The physical result of this cascade? Irreversible paralysis of the leg muscles.
That brings us to case file number 4, cyanide in the diet. Let's discuss cassava toxicity.
It is actually a startling reality that hundreds of millions of people rely daily on a staple crop that is naturally armed with cyanide. I'm talking about cassava. For the food technologists in our audience, you already know cassava root is a vital source of dietary carbohydrates globally. But the proper processing of this root, the careful chopping, the grinding in running water, the fermentation, that is literally a life or death science. These steps are absolutely required to trigger the enzymatic release and the washing away of hydrogen cyanide before the food can be safely eaten.
And this brilliantly illustrates the intersection of all our disciplines.
From the food tech perspective, we absolutely must hydrolyze the lamanarine to safely remove that toxin. But what happens when that vital processing fails, or maybe it's rushed due to severe economic hardship? Well, that is exactly when our clinical scientists see the tragic results. Chronic, low-dose exposure to cyanide, particularly in a nutritionally poor diet, leads directly to tropical ataxic neuropathy, or TAN. These patients suffer from depleted sulfur amino acids, leading to severe nerve atrophy, deafness, and ultimately optic nerve blindness.
Now, for those working in acute toxicology, the acute molecular mechanism is incredibly swift. Cyanide binds directly to the ferric ion of cytochrome oxidase right inside the mitochondria. This completely halts the electron transport chain, which triggers an immediate cessation of cellular respiration. The cell literally just suffocates. Fortunately, we do have antidotes like hydroxycobalamin, which works by binding the cyanide incredibly tightly, allowing that whole complex to be safely excreted in the urine.
Finally, case file number 5, the lethal lectin. We are talking about ricin.
So, we are moving away from chronic dietary exposure now and into acute, highly lethal toxicity. Did you know that just 5 raw castor beans contain enough of the toxin ricin to kill an adult human within a matter of hours? It is genuinely one of the most potent plant toxins known to science, and obviously a primary concern for forensic and clinical toxicologists everywhere.
Ricin is essentially a 60-kilodaltoning glycoprotein assassin, and it works through a really clever 2-part mechanism. Think of it as a lockpick and a saboteur working together. The B chain is the lockpick. It binds to the cell surface carbohydrates and facilitates entry right into the cytosol. Once they're inside, the A chain acts as the saboteur. It irreversibly inactivates the 6DS ribosome by removing just a single adenine residue. But that is enough. This completely halts all cellular protein synthesis, and the cell dies. Notably, though, simple heat deactivates this protein entirely, which is exactly why commercially processed castor oil is perfectly safe to use.
To bring all of this together, let's just reflect on this quote from the literature. Whether you're a plant scientist analyzing the soil, a food technologist processing these crops, or a clinical chemist treating the patient at the very end of the line, we are all observing the exact same phenomenon. We are constantly navigating this ancient, microscopic evolutionary arms race between plants and animals.
I really hope this explainer has provided a valuable, cross-disciplinary perspective on these incredibly powerful molecules. So, I will leave you with this final, provocative thought. Next time you prepare a meal, ask yourself, what microscopic chemical warfare are you disarming? Thank you so much for watching this explainer.